يلا. بسم الله الرحمن الرحيم. السيد معاون العميد المحترم، السيدات والسادة رؤساء الفروع المحترمين، أساتذتي، زملائي، الحضور الكرام، السلام عليكم ورحمة الله وبركاته. إنه من دواعي سروري أن ألتقيكم اليوم حتى نقدم لكم إن شاء الله نبذة عن بحث التخرج اللي قمت به بالمملكة المتحدة من أجل الحصول على شهادة الدكتوراه بالصيدلة. The project title was Personalized Warfarin Dosing in Children after Congenital Heart Surgery using the model-based approach. First, I would like to give you a brief background about the issue behind doing this project. Traditionally, drug dosing in children has been usually extrapolated linearly from adult values, and the doses are adjusted mainly according to body weight or body surface area. But as you know, children are in a continuous state of development and maturation, and this causes significant inter and intra-individual variability on the drug's pharmacokinetics and pharmacodynamics, and this has well been established. In addition to that, there is the effect of genetics on both pharmacokinetics and pharmacodynamics. Therefore, this simple linear extrapolation from children doses may lead to inequivalent exposure and or response in the two populations. Therefore, in order to optimize drug therapy and hence improve treatment outcome, knowledge about the drug pharmacokinetic and pharmacokinetic parameters for the individual patient is essential. The concept of personalizing drug dosing in children using the Bayesian forecasting recognizes every individual as has unique pharmacokinetics and pharmacodynamics that control the time course of the drug effect. The Bayesian forecasting is a proactive approach to dose individualization of drugs with narrow therapeutic index. This method utilizes population PK/PD models that incorporate the variables that explain the inter and intra-individual variability in order to prospectively predict the individual patient's PK and PD parameters and then to optimize drug dosing. Now we come to the drug under investigation, which is warfarin. As you know, warfarin is the most widely prescribed oral anticoagulant. It represents a major challenge in drug therapy in children. The main indications for warfarin use after congenital heart surgery is long-term anticoagulation. Alaykum as-salam wa rahmatullah. Long-term anticoagulation after Fontan procedure, and also in case of mechanical valve replacement. To give you a simple idea, a brief idea about Fontan procedure, the Fontan is the last stage of a series of procedures done to children born with single functioning ventricle. What they do is that they direct the blood from the venous system directly to the lung, and they keep the single functioning ventricle for pumping blood to the systemic circulation. So these patients are called patients with Fontan or with Fontan circulation. They require lifelong anticoagulation with warfarin. As you know, warfarin is a neurotherapeutic range drug. It requires accurate dosing and frequent monitoring of the international normalized ratio, INR, to prevent both over anticoagulation and under anticoagulation. Both of these lead to either bleeding or thrombosis, so both of them are life-threatening condition. This is very briefly the mechanism of action of warfarin. Warfarin is vitamin K epoxide reductase inhibitor. Inhibit this enzyme that is responsible for the production of reduced vitamin K that is in turn required for the activation of clotting factors 2, 7, 9, and 10, and the anticoagulant proteins CS and Z. Warfarin is available as a racemic mixture of R and S isomers. The S isomer is 3 to 5 times more potent than the R isomer as anticoagulant. Therefore, most of the studies focus on S-warfarin and its metabolism, the genetic polymorphism involved in that, more than the R isomer. S isomer, more than 90% of the S-warfarin is metabolized by CYP2C9, and we will come now to the genetic polymorphism of this enzyme. Warfarin, there are many factors that contribute to the variability, inter-individual variability of warfarin PK and PD. So many factors. These, like, for example, age, if there is comorbidity, drug-drug interaction, adherence to treatment, the coagulation system itself varies between individuals. For children, variable intake of vitamin K, especially for those lower than 1 year, they have the formula milk that contain vitamin K. Children especially are more prone to frequent illnesses, antibiotic use, the formulation used in children. All these are factors contribute to the variability in warfarin PK and PD. In addition to the effect of pharmacogenetics of CYP2C9 and VKORC1. CYP2C9, the enzyme responsible for the metabolism of most of S-warfarin, has genetic polymorphism. The two most common polymorphisms in this enzyme that produce reduced metabolic capacity of the enzyme are designated as star 2 and star 3. And as you can see, patients who have variant alleles of this enzyme require dose reduction. So especially those that have 2 copies of star 3 require around 80% reduction in warfarin dose. The same thing applies for VKORC1, the gene that encodes the enzyme vitamin K epoxide reductase. So there is also genetic polymorphism in this enzyme. So people who have genetic variants of this enzyme require about 25 to 50% dose reduction of warfarin. Now we come to the model that we have used in our study, the Hamburg model. The Hamburg model is a PK/PD model for warfarin dose individualization. This model can predict the dose that can give the most, reach a given target INR, and also predict INR for a given dose. This model can be used for the prediction of both initial and maintenance doses of warfarin. This model was developed in Sweden by a Swedish author, Anna-Karin Hamburg, with her colleagues. It was first developed in adult Swedish population, and then it was bridged to children and further optimized for use in children. This is a schematic presentation. So as you can see, the PK model and PK model and PD model, and this model is the transduction, because you know there is a lag phase between administration of warfarin and the increase in INR, because warfarin acts on the synthesis of clotting factors. It doesn't have any effect on the factors that are present in the circulation. Now, optimizing a drug therapy is important. However, adherence to this prescribed dose is equally important. So if I prescribe a personalized dose and the patient doesn't take that dose, there's no point of what I'm doing. So usually patients in general and children as well, the same with their families who are on lifelong treatment, they are required to adhere to a specific regimen, especially like this with warfarin. They have to restrict vitamin K intake. In the Western population, they have to restrict alcohol intake. They have to take care about any kind of drugs that can cause interaction with warfarin. In addition to remember taking the dose every single day, and then monitor the INR at specified intervals. So adherence in this case is very important. There is a range of factors that can affect adherence in children, such as age, family factors, socioeconomic status, and so many others that can affect adherence. Therefore, understanding the experience and the views of families as well as health care providers about how they monitor or how they deal with warfarin is important, and also obtaining their views and their experience with this new dosing approach is also important. The aims of the study. The first aim of the study was to validate the Hamburg model in the population and our population in our study in United Kingdom. The second aim was to prospectively compare warfarin dose management using the PK/PD model as compared with the traditional trial and error approach. The third aim was to explore the lived experience of families and healthcare providers with managing warfarin treatment, as well as their experience with the new dosing method. This study was conducted at the East Midlands Congenital Heart Center that is located at Glenfield Hospital in Leicester. This is a tertiary hospital that covers the East Midlands area. This area presented as dark green in the map. So as you see, this area is composed of several counties. So all patients, children with congenital heart disease from these areas are referred to this center for surgery and then afterwards for follow-up. So, study design and the procedures. The first part, this study can be separated into three parts. The first part was a retrospective validation of the Hamburg model. So as I have said, this model was developed in Sweden. So we wanted to know, is it valid for use in the UK? Is it the same? Can it work on that? So this retrospective validation we obtained, we have taken data from the database of the EMCHC, that center, and we have estimated warfarin doses for these patients and compared them with the observed or actual doses that were administered to the patients. The second part was a prospective interventional study to assess the new dosing approach in two groups of patients. The first group was patients starting warfarin for the first time after congenital heart surgery. In those patients, mouth swab for genotyping of CYP2C9 and VKRC1 was taken, and then initial and maintenance doses of warfarin were estimated by the model, and INR monitoring also was all done, was interventional. These patients were followed up for 6 months, and afterwards they were compared with historical case-matched controls. The second group included patients established already on warfarin treatment, on long-term warfarin treatment. Those patients were entered a randomized crossover trial in which they were randomized to either model phase, where warfarin doses and INR monitoring was done by the model, or the doctor phase, where warfarin management was done by the doctors as per the usual clinical practice. Both phases were followed up for 6 months. Afterwards, the patients were crossed over to the alternative phase of treatment and followed up for further 6 months, and then we compared the two phases of treatment. The third part of the study included exploring the experience with managing warfarin treatment. So in order to do that, we have conducted semi-structured interviews with subsamples of families and doctors and nurses to obtain their views about managing warfarin treatment, as well as their views about the new dosing approach. The study outcome measures. For the retrospective study, the outcome measures included model accuracy. This we meant that the difference between model predicted doses and observed doses, bias and precision. The second outcome measure was the clinical accuracy. This included the patient percentage of patients with ideal dosing within 20% of the observed dose, under predicted and over predicted doses. For the prospective study, the outcome measure was to compare the two phases of a treatment, the model phase and the doctor phase, in the following parameters. For group 1, time taken to achieve the first therapeutic INR, time taken to achieve stable anticoagulation, time taken to over anticoagulation. For group 1 and group 2, this included the percentage of INR measurements within the target range. This was represented as percent ITR. The percentage of time in target therapeutic range calculated by linear interpolation. This is as represented as percent TTR. Frequency of INR measurements per patient per month. Frequency of dose alterations and the number of INR values above 4 and 5. The outcome measure for the qualitative study included the experience of healthcare providers and families about managing warfarin treatment and the new dosing approach. This is the model, as I have shown you schematically, was converted into a Java-based software. This is how it looks. For the initial dose prediction by the model, we need to input data about the patient's age, weight, CYP2C9 and VKORC1 genotype, the target therapeutic range, and baseline INR value. For the initial dose prediction, the model uses the population data and the individual patient covariates, the age, weight, and genotypes to predict the initial dose for that patient. Once we get the INR feedback from the patient, we need to input that into the model. And as you can see, there is a specific format for inputting data into the model. This includes the date, time of the dosing and INR monitoring, and weight of the patient. From this data, we can estimate the individual patient PK and PD parameters, K10, the elimination rate constant, and EC50, concentration required to produce 50% of the effect. As you can see, the model also predicts the INR, this black curve of the population, and the individual patient INR, the red curve, and also you can see the observed INR values for the patient. By looking at these observed INR values, you can assess the individual fit of the curve so that you can get the best dose predicted. From that, depending on these parameters, the model will estimate the maintenance dose or make the dose adjustment. Genotyping of CYP2C9 was done by para-DNA technology. This is a point-of-care technology. We take a mouth swab from the patient, transfer that to a reaction plate, to the machine, and we obtain the result within 45 minutes. This technology was really important, especially for children who are starting warfarin treatment, because we need to obtain the results as soon as possible. Ethical approvals for the prospective study. The prospective study was first approved by the Ethics Committee of De Montfort University in March 2015. After that, it was submitted to East Midlands Research Ethics Committee, and the approval was obtained in September 2015. After that, the study was approved by the Research and Innovation Office at University Hospitals of Leicester in October 2015, after which patient recruitment started. Results. For the retrospective study, data from 87 patients was collected. Only 60 patients had complete data of warfarin dosing and monitoring right from the beginning of treatment, so they were included in the evaluation of the model. The age range was from 1 to 15 years, around 16 years. Most of the patients were male. Around 70% of them were of white ethnicity. The major indication, around 70%, is Fontan procedure. And in addition to AVR, this is aortic valve replacement, and MVR, mitral valve replacement, in a lower percentage. Ideal dosing was 70%. and 25% of the doses were underpredicted. The bias was minus 0.1. This means that there was a tendency of the model to underpredict doses by 0.1 milligram. Precision was 0.19. This means that there was an imprecision of 19%. And as you can see in this figure, the relationship between observed and model predicted doses. For group 1, 5 patients were enrolled over a 14-month recruitment period. All of the age range was from 5 to 9 years old. All of them were female. 3 were Asian, 2 were white ethnicity. All of them were of the wild type, CYP2C9, which is star 1, star 1. 3 of them had the wild type, VKORC1, and 2 were heterozygous, AG. 3 of them were Fontan patients and 2 mitral valve replacement patient. Case control matching was done based on age, indication, and target INR range. In these figures, because of the small number of patients, the results were demonstrated descriptively. In this figure, time to first therapeutic INR was longer for the case subjects. This is, as I said, because of the method of initiating warfarin treatment. The usual clinical practice, they start with loading dose of warfarin, 0.1 to 0.2 milligram per kilo, whereas for the model, it uses population data adjusted with the patient and give the first dose. In addition, we had one of our patients had issue that she was started on a lower target range because of a risk of bleeding, and then this range was increased. So this has led to this kind of results. Time to over anticoagulation was longer in the case subjects. This is something positive for the model, that the time to over anticoagulation is longer than for the case subjects. This also may be due to the loading doses that can lead to overshoot of the INRs out of the range. Time to stable anticoagulation was shorter for the case subjects. They reached stable anticoagulation faster than the controls. For the time in therapeutic range, both measured as percent ITR and percent TTR, both of them were higher for the case subjects as compared to the controls. Number of dose changes was comparable in the 2 groups, and the frequency of INR measurements per month was also comparable. The INR above 4 and above 5, the median was comparable. However, the number of patients we had 3 of our cases with no INR above 5 as compared to the controls. All of them had INRs above 5. For group 2, 29 patients were enrolled in the study. 26 patients were included in the final analysis. Age range was from 1 to 17 years. Mostly about 70% were male patients, more than 75 white ethnicity, CYP2C9, more than 60% wild type. VKORC1, we have 46 of the wild type, 53 of GA, heterozygous for the variant allele. More than 75% of the population were of Fontan procedure, and we have 6 patients with MVR and AVR. For the study outcome measures, the percent ITR and percent TTR for both phases of a treatment was not significantly different. However, we have had 5 patients who experienced medical issues in one of the phases of a treatment. So we have conducted a sensitivity analysis and excluded these 5 patients. When we have conducted this analysis, the percent of time in therapeutic range for the model phase was significantly higher than that of the doctor phase. For the frequency of INR measurements, also was not statistically significantly different, but for the frequency of dose alteration, it was significantly higher for the model phase. This again was because of the method of dose estimation. The model uses the mid of the target therapeutic range and adjusts the dose to that value. While in practice, we will come to this soon, it's something different. This is the time in therapeutic range for the cases that we have excluded from the analysis. Patient 1 had his medical issue in the doctor phase. The 4 others had their medical issues during the model phase. For the INR values above 4 and 5, it wasn't significantly different. However, you can see that the model phase had lower patients with maximum number of INR values above 4 and 5. We have also done some subgroup analysis to compare the time in therapeutic range between the two phases of treatment. The subgroups included the indication for warfarin, the age, and the genotypes of CYP2C9 and VKORC1. As you can see, favoring the model phase is on the right-hand side, favoring the conventional approach is on the left-hand side. For patients with Fontan procedure, after excluding the 5 patients with medical issues, the time in therapeutic range was significantly higher than that of the model phase. The effect of genetic and non-genetic variables on warfarin dose calculated as milligram per kilo per day. The patients with variant allele of VKORC1 required significantly lower doses than those with the wild type genotype. Now we come to the qualitative study. As we said, we were exploring the experience of doctors, nurses, and families with warfarin treatment. Interviews were conducted with the 3 doctors, 2 nurses, 2 group 1 families, and 2 group 2 families. These interviews were recorded digitally, transcribed verbatim. What we mean by transcribed verbatim is that the whole interview was written word by word, just exactly the same way that it was said by the participant. And then these interviews were all analyzed to see what are the themes that can emerge from them. For the doctors' interviews, 3 themes emerged from them. 1, the medical and the clinical knowledge. The doctors in their interviews said that the key determinants of warfarin treatment in children after congenital heart surgery were the indication for warfarin and the clinical condition of the patient. They said that patients with mechanical valve replacement were more concerning to the patient, to the doctors or to the whole staff than those with Fontan and even than those with aortic valve replacement. They required special attention because of higher risk of thrombosis. Guidelines for dosing and monitoring were available, but they can individualized according to the patient's condition. There was variability in prescribing warfarin between the doctors as well, depending on the personal experience of the doctor. Maintaining therapeutic INR levels was challenging in children less than 5 years of age and also in adolescent patients. Less than 5 years because we said infection, antibiotics, use of formula milk, all these things. For adolescents, the adherence to taking the drug, in United Kingdom, the issue of alcohol intake. So they consume large amounts of alcohol, so all of that affects warfarin control. For the INR monitoring, the doctors stated that patients with Fontan circulation required less frequent testing and wide range of INR values was accepted above and below. We said that the model adjusts the dose to the mid of the target range, while in practice, for example, if we have a target 2 to 3, the doctors adopted a range from 1.5 maybe up to 4, wider than the actual target INR range, because they say that it is acceptable and there is no risk of bleeding or thrombosis, minimal risk. patients with MVR, mitral valve replacement, they require more frequent testing. Low INR levels are very concerning because they are associated with increased risk of thrombosis. And when the INR levels go low, they admit the patient to hospital for intravenous heparin or maybe for the low molecular heparin. So this was not convenient both to the hospital and to the families as well. We have also noted we do a clinical trial when the target range is 2 to 3. If it is 3.1, so it's out of range. What happens in a clinical practice is different from the literature. The doctors were very cautious about reducing or stopping warfarin treatment when the INR goes very high, because they said that they have experienced rebound drop in INR if they stop warfarin treatment. So they prefer to slightly reduce the dose instead of stopping it altogether. The dosing was challenging to the doctors in patients who where very small dose changes produce significant INR changes. Those were challenging to the doctors, as well as young children, very young children, those like below 1 year of age with very small mechanical valves. The dose adjustment is like 0.1 or 0.2 of milligram. So the accuracy of dosing, the response to this dose change is very critical. Experience of the nurses. The nurses talked about their role in the process of warfarin dosing and monitoring. It was mainly about education about warfarin and the use of the home INR monitoring machine. Children are usually monitored by home. This is the home machine, just like the glucometer, by a strip, a drop of blood, and you get the INR. Then they ring the hospital to report this INR value, and then dose adjustment is made accordingly. The nurses also reported that the major forms of non-adherence included non-adherence to the prescribed dose. They said that there is a few families that do their own dosing. They do not listen to what doctors say, and they say that we know our children better than even the doctors. And we have had one family of them that I will come shortly to them. Also missed dosing, increased intake of vitamin K diet, also excessive alcohol consumption by adolescent patients. These are the major forms of non-adherence. Families. When we have interviewed the families, they said that the period at the beginning of treatment of warfarin treatment was concerning to them because of the new drug administered to them that requires special attention. So this was challenging to them and worrying, but they were provided with information, and then families gradually started to adapt warfarin to their daily life. The concerns raised by the families was the easiness of bruising. So this led to a restriction of sports. For young children, they have to be careful if they hit something because they can very easily bruise. Also for females, the families were worried about menstruation and pregnancy in the future, and also about alcohol restriction. For the dose decision, 3 of the families interviewed preferred that a professional with warfarin dosing does the dose decision for them. In contrast, there was one family that said the best judge of dosing warfarin are the parents, because they live with their child. They know if the child is not eating well, if the child feels unwell. So all these things, they know them better than the doctor. They say that the doctor is not with me at home, so I am the best judge. And they were confident in their dosing by 99%. Adherence to treatment. Several factors were recognized as affecting adherence to warfarin. This was first the importance of the drug itself for the medical condition. The patient's age. We had, children from 1 to 18. So small children are completely reliant on their parents in their dosing and monitoring, as compared to adolescents who start to be self-dependent and they do their own thing. The treatment regimen. The frequency of dosing was convenient for them, once daily dose for warfarin. So they adjusted to a particular meal, a particular time of the day. The dosage form, some families of older children preferred tablet dosage form, while families of younger children said that liquid dosage form is more convenient. And the home INR monitoring, having the home INR monitoring machine was very convenient for families, especially those who live outside Leicester, because this decreases the cost of traveling and time taken out of work and out of school. The family role was very important in the process of managing warfarin, and also the relationship with healthcare providers. The experience with the model. The doctors and nurses stated that the model-based dosing was useful and acceptable in patients with stable medical conditions, mainly those with Fontan and maybe those with AVR, aortic valve replacement. They said that the model-based dosing was consistent and helped to reduce the inter-individual variability in doctors' dosing, and it was faster to the patients than the usual daily process of monitoring, of prescribing. However, the doctors suggested that the model-based dosing is accompanied by clinicians' judgment in patients who have some medical complexities, and the use of target INR ranges similar to what they use in a clinical practice, instead to sticking to a given target therapeutic INR range. Both doctors and nurses prefer to have more experience with this new dosing approach and to wait for this study's result before recommending its use in a clinical practice. For families, again, 3 of the families said that the model dosing was consistent, the INR control was stable, except the one family that was not happy with the model dosing, because obviously they wanted to do their own thing. They said that parents had equal responsibility with doctors and nurses in managing warfarin treatment for their child. So they were not happy. They gave negative comments about the model. Conclusion. This research project has extended the current knowledge on the clinical application of model-based genotype-guided dosing of warfarin in children after congenital heart surgery. The new dosing approach can improve the anticoagulation control in children. This is most likely in children who start warfarin for the first time after surgery and those with Fontan circulation. The study has demonstrated an overall acceptance of healthcare providers with a new dosing approach, and it has provided an in-depth understanding of how warfarin is managed both by the healthcare providers and families, and also an understanding of both families and healthcare providers of managing warfarin treatment. However, because of the small sample size of the study, a multi-center randomized controlled clinical trial is required to evaluate the clinical effectiveness of this dosing approach, in addition to a pharmacoeconomic study to evaluate the cost effectiveness of applying such dosing approach in a clinical practice. Thank you so much for listening, and sorry to be that long. Thank you.
Question about the phase 1. What's the reason behind using like 5 participants only in phase 1?
Because it was like a group 1 start for the first time. Because according to literature, the effect of genetics is more pronounced during the start of warfarin treatment, during the first 3 months of therapy. So we wanted to see if the model-based approach produces better control for them.
Did you do any statistical analysis for this?
No, just descriptive statistic. This was because a very small number of patients that presented for surgery that required warfarin treatment afterwards. So like we had to wait like for 3 months to get 1 patient enrolled in the study.
Can we call the second, like the interviews, like qualitative?
It is qualitative research.
Qualitative. And
it is.
You use the shared common themes to find the findings?
Thematic analysis. This is a special field and special theories about behind that.
Okay, thank you so much.
Yeah.
this study applied or experienced in our country when you came by?
Well, as it is for warfarin, I'm not quite sure if we have children kept on warfarin treatment. That's one. But the idea or the concept of personalized dosing is important, yes. It's not only for warfarin, it's all for drugs with narrow therapeutic range, like for children, anti-cancer drugs, antibiotics with narrow therapeutic range, like for example, vancomycin. So it is really important. We need to take this step forward.
But you didn't do anything.
Not yet. I have just arrived.
Try.
Yes.
خوش يعني محاضرة دكتورة، بس بالنسبة لهنا يعني المشرف مالتك صيدلي ولا طبيب؟
صيدلي PhD في فارماسي يعني بالمستشفى، لأنه أنا كان عندي 3 سوبرفايزرز، أكيد اللي بالكلية بالجامعة من سكول اوف فارماسي أكيد، واللي بالمستشفى أكيد يعني PhD فارماسي، وأصلا هو يعني فارماكوكاينيتيست أصلا يعني بالباك جراوند از ويل.
زين هنا يعني اشوف بالسلايدات كلها شنو دور الصيدلي هناك كلينيكال؟ انت اخذت اختصاص كله دكتور نيرس
يس فور وارفارين يس فور وارفارين
يعني البحث منحى طبي يعني ككلينيكال فارماسي يعني شنو الدور يعني كانت كلينيكال فارماسي؟
You need to adjust the dose. Who does personalized dosing؟ يعني خلال الفترة هاي مال الدراسة كلها نعم
الاطباء هنا بالعراق
احنا اللي الدراسه سويناها باليونايتد كينجدم وقدمنا ريكومنديشنز هناك، فهنا لما تجي مثل ما قلت للدكتوره هنا مو شرط الا الورفرين نفسه، عندك انت وايد رينج اوف دراجز وذ نارو ثيرابيوتيك اندكس، عندنا هوايه اطفال مرضى الكانسر، هذا كلش ابليكابل ان ذيس فيلد. وهسه التوجه انه include pharmacogenetics يعني حول العالم انه you have to include pharmacogenetics in your dosing, in your diagnosis, in your even for cancer patients if there is a future risk for this family member to develop cancer if they secrete the genes and you find in the family history that there is a predisposing gene for cancer. So كل التوجه هسه بهالاتجاه. ف it's really important.
You can see the prospective for.
نعم دكتور.
Is it a routine practice in UK to perform pharmacogenetic analysis for any patient taking one?
No. This was for the first time. فلهذا لما سوينا برزنتيشن في اكثر من مكان بمستشفى للاطباء سالوا عن الكوست مال هذا الجهاز، ايش قد الايجار وايش قد الكت تكلف for each patient.
يعني generally
for them لا convenient يعني for the prices in the UK it's convenient for them. يعني الكت الرياكشن بليت 25 باوند 25 باوند بالاضافه الى الايجار مال الماشين نفسها مانثلي ريت.
هذا كيف اثناء السلايدات والله يعني في ستريت لاين طلع عندك هذا شنو السلوب مالته هذا هيك يعني
هذا لا كومباريسن
according to conditions
لا هو هذا سويناه يعني على مود بس نعرف انه الكورليشن بتوين ذم يعني بين البريدكتد والاوبزرفد دوسز.
نعم
زين احنا ما عندنا الفاسيلتي اللي طلع لك اي نوع من الانزيم سوري مال البوليمورفيزم بالنسبه للانزيم راح يسوي لك ميتابوليزم اي احنا ما عندنا اصلا فاسيلتيز اللي تطلع لك اي
الجينيتيك بوليمورفيزم اوف ايتش بيشنت اي بالضبط اعتمادا على البي سي ار فلازم سامبل ويودوها لللاب. اي هو حتى باليوكي احنا من سوينا هاي الدراسه هو كان في شيء جديد يعني هذا الماشين يستعملوها بالفورنسيك مال الجريمه بالكرايم بليس ياخذون السامبل وعلى مود يسووه فطوروه وسووا جزء منه على سب 2 سي 9 في كي ار سي 1 واعتقد على سب 2 دي 6 هذا اللي هو امبورتنت فور كودين از ويل فيعني جديده.
يعني الميتابوليزم عندك حتى الاكسكريشن الكونجوجيشن من الباثوي هم اللي يختلف للاطفال عن الكبار. اكيد هذا لازم نسويه اكيد. احنا بالاطروحه مالتي يعني كلها حطيت يعني من الفيسز من الابزوربشن الى الاليمينيشن شنو الديفرنسز بتوين ذم بين الادلتس والتشيلدرن اكيد يختلف. نعم دكتوره.
بالنسبه للوايلد تايب ذكرتي انه هو الجي جي منين اخذتوا الريفرنس له؟
فور في كي ار سي 1 هو اكو هوايه الريبريزنتيشن مالتهم واحد يقول تي سي واحد يقول جي جي بس من ترجعين تقريها يقول they are in سترونج ديسإكويليبريم، فمعناته اذا اكو واحد من عندهم موجود فمعناته الثاني اكيد موجود. فاحنا بالكت مالتنا يقيس الجي جي جينوتايب، فاخترناها انه تبقى كلها تمشي على الجي جي.
هنا مثلا بالعراق اكيد حدثت الوايرد تايب مثلا ديزيز معين او في جين معين اقصد.
اكيد. فانت لازم تعرفين شنو الجينوتايبس الموجوده، يعني هسه مثلا عفوا سب2C9 اكو ب 60 جينيتيك بوليمورفيزم، بس الموست امبورتنت اوف ذم هم ستار 2 وستار 3 اللي هي تسوي ريديوس بالميتابوليزم، ريدكشن significant reduction in metabolism. نعم.
ولو قليله واكثر متجه تجاه كانسر يعني خاصه
كلش مهم طبعا اكيد.
لكن مثلا هذا نفس الموديل مال البروجكت يعني يا ريت مثلا سامبلات خلي تنأخذ من مستشفيات الاطفال على نفس التايب طريقه ايضا نفس الشيء وايد تايب مثلا بنفس الجينات حتكون اسهل تقريبا الاجهزه موجوده مثلا مراكز بحثيه.
والله انا اتمنى، بس لانه هو انا عندي توني رجعت فبعتني دا استكشف الوضعيه.
الجهاز اللي اشتغلتوا به جينوتايبنج
نعم.
Personalised for Warfarin لو اني جينوتايبنج.
مو اعتمادا على الكت اللي انت تاخذها والسوفت وير. هو البيسك مالته هو PCR. بس يعتمد على انه مو بدل ما تاخذ بلاد سامبل هي سواب وتسوي اي اكو اي
هذا لا يعني البيئه
اي بس ما يسوي
الرياكشن بليت بالبوتم مالته اكو كيميكالز فهي اللي راح تحدد الجينوتايب، واكيد اكو سوفت وير معين يربط بالجهاز ويحدد لنا نوع الجينوتايب اللي يطلع.
يعني دايركت الجينوتايب
اي دايركت راسا يطلع لك 45 دقيقه سب2C9 كذا جي VKRC1 كذا دايركتلي.
طيب اكو جينوتايبنج مالت بالنسبه للوارفرين وكذا موجوده يعني مثل
اي بس التكنيك دكتور يعني لازم تسحب بلاد سامبل وتوديها للمختبر. اي اكيد طبعا اكيد يعني هو وورلد وايد هذا مال مساله الجينيتيك بوليمورفيزم اللي يخص الوارفرين كل مكان. نعم دكتور.
First, congratulations for the PhD.
Thank you so much.
Second, thank you for your interesting talk. We really enjoyed it.
Thank you.
I have a couple of questions. First, what's the difference between genotype and phenotype?
Yes.
And second question, what are the challenges for application of personalized therapy? I mean, I'm talking about big companies.
Okay.
So far, I don't think there's like any kind of personalized like therapy or drugs for special population of people. So, what do you think the challenges behind this?
Challenges in Iraq or challenges elsewhere?
I mean, I'm talking about global application.
Yes. So for the first question, genotype and phenotype. Genotype is what your genetic map is. So in your genes you have this sequence of, for example, your eye color. Phenotype is what is the color of your eyes. How your body metabolizes the drug. How much of the dose you need for that drug. This is the difference between genotype and phenotype. Challenges for such thing, first you need to put this software incorporated into an electronic system first, because you need to get the results, put them into the software, analyze and get the dose. So it needs to be done in a timely manner. Now the approval. First, you need to do studies, randomized controlled trials to approve that this is beneficial. There is significant difference. That is one. As I said, for warfarin, the difference between a clinical trial, we are very strict in like target ranges 2 to 3. In practice, it's something different. So the software needs, for my study, the software needs to be improved so that you can meet the clinical needs. It needs to be fast, needs to be approved. So these are, and you need personnel who will work on that software. يعني for me it was me 24 hours responsible for this. So at home, weekend, whatever, ring me from hospital, INR, we need a dose. So this was really exhausting. When you do this in clinical practice, you need someone available all the week, or if the drug, like for warfarin, it's like daily. It's continuous, not like for a specific period of time. So this is the personnel who work on this also important. شكرا. Any other question? شكرا جزيلا. أسعدني حضوركم. شكرا جزيلا.